So we left off last Thursday talking about different types of eukaryotic cells. We'd introduced some of the things that are common between all living things, like a plasma membrane, and we talked about prokaryotes and archaeas, and now we're ready to really get to the meat of the rest of the class, which is the eukaryotes. So we've got a couple of eukaryotes up here, animal cells and plant cells. Both of these are members of the eukaryotic kingdom. And just a couple of things to note, some of the things we talked about. You see a lot of common organelles between these two. Both have a nucleus. That's part of being in the eukaryote club. Both have rough ER, smooth ER. Mitochondria are present in both. Golgi are present in both. The only thing you really see different between these cells is over in your plant cell, it's got a couple of extras. So same thing as an animal cell, except more in your plant cell. So you've got a large central vacuole involved with pH maintenance, water maintenance, kind of a waste cleanup area for a plant cell. We also see those large green chloroplasts, and those are going to figure pretty prominently coming up in Chapter 5. So we'll introduce them here, but we'll really kind of talk about their structure and function more in depth in Chapter 5. We also see on this one a cell wall, but note that the cell wall is just on top of the plasma membrane. The membrane is still there, it's just underneath the cell wall. The cell wall just gives us some strength, some rigidity, helps with water regulation, so it's there but on top of the plasma membrane. So if we look at our next slide, we start talking about the nucleus. I mean, this is what makes a eukaryote a eukaryote. This is the club, is to have this central control center of the nucleus. Now within this, we're going to find our primary complement of DNA. And I really kind of urge you to hit on that primary complement of DNA. When you look at a skin cell, if you look at your eyes and there's cells making up your eyes or your hair, any of those features, you got 50% of that from mom and 50% of that from dad, and that's your primary complement of DNA, that inherited genetic material. If you get a cut on your hand and you want that to heal and you want more skin cells, you're going to replicate the DNA there, and that's DNA that's being replicated is the stuff in the nucleus. So this is your primary inheritable genetic information. Now in a eukaryote, because we have organelles, because we have the ability to kind of increase surface area so we can do some snazzy things, our nucleus is kind of really under lock and key. We have a very special membrane structure on this. Remember when we talked about looking at a cell underneath a light microscope, we would see the nucleus. But we'd have this cytoplasm, stuff we really couldn't see all the organelles in. So we just kind of characterized it as the stuff from the plasma membrane up to the nucleus. When we get up to the nucleus, our membrane is quite different from the membranes we find around other types of organelles. It's actually two layers, two lipid bilayers there around the nucleus. But we still have to get stuff in and out of the nucleus. So we actually have some large pores for moving things in and out. And some stuff that needs to go out. Remember our kind of central dogmatic idea here of information flow is we've got DNA as our blueprint, RNA as our contractor, and that has to go out and make a house, which is our protein. Well, the RNA's got to get out of the nucleus to be able to make that protein. So RNAs are obviously exiting out of these nuclear pores. It's a pretty big molecule, so we've got to have some room to get that out. But we also have stuff coming back in. And if you think about it, all that stuff that's going on inside that nucleus, anything that's happening, that's being done, what's the what's our molecule there that's doing action, that's causing an event to occur? If I'm going to say that I replicate the DNA, what's doing that replication? And hopefully you're thinking of a protein. Proteins do everything. If something's being done or is happening, golden answer is protein. So we obviously need proteins in there, but our proteins don't get made in the nucleus. That RNA exits out into the cytoplasm, that RNA meets up with a ribosome, we put together a protein out there in the cytoplasm. some of those proteins need a flag to come back to the nucleus. And so if they've got the right flag on them, they'll be able to enter in through those nuclear pores, and they have a vital role, kind of synthesizing new DNA, helping to make RNA, being able to scaffold the DNA so it'll actually fit in that nucleus. There's about 2 meters worth of DNA in that nucleus. And that's every single cell you have. Every single skin cell, every single liver cell, every single lung cell, every single brain cell has the same exact DNA. And it's about 2 meters in length. So it's got to be pretty compacted into that nucleus. And that, having some action done, is going to be a protein that does that. So we've got to get proteins in there. Also, when we start talking about making more cells, well, the example I said a second ago, if I get a cut on my hand and I want new cells so that that cut heals, I need to replicate the DNA because I want those cells, those new cells, to have exactly the same DNA as the old cell. If I don't have exactly the same DNA, I'm going to have some problems probably. So I want exactly the same DNA. But I want that DNA to be replicated. I want it done right because I don't want to get some sickly cell out of this. I want it to be done properly. If I want it done properly, the most controlled place for that to happen is inside the nucleus. So when DNA is synthesized in the context of that cell is going to divide and make more cells, that's going to happen inside the nucleus as well. So from our nucleus, we've got our DNA, and kind of another thing to notice is in the middle of that nucleus, we've got a dark spot. Even in light microscopy, a lot of times you'll see that dark spot inside the nucleus. That's called the nucleolus, and that's pretty important because that leads us to talking about our next organelle, if we're going to call it an organelle. Remember, this is kind of a gray area. Our next organelle that we're talking about is the ribosome. And whether it's an organelle or not, it's kind of a gray area. Remember, it's not membrane bound, but it is a structure that's carrying out activity, kind of division of labor stuff within the cell. What it is just a bunch of RNA and protein wadded together. If you think about it, that makes sense. If you think back to our discussion last time, we talked about our nucleic acids, and we needed to take DNA and RNA, which were both nucleic acids, and we needed to make them into a protein, which was an amino acid. Now, how did we do that? Well, we had a code for that, the codons. The codon is switching us from one language to another language. If you look at the ribosome, that's where that switch is occurring, and both languages are there. There's both nucleic acids making that thing up and proteins making that thing up. So we've got our ribosome, but it's made out of RNA and some proteins. If we look back at that dark spot inside the nucleus, the nucleolus, actually that's where a lot of the pre-manufacturing occurs for our ribosome. So we start kind of prefabbing some of it together and then ship it out of the pores. It goes out into the cytoplasm and finishes getting made. So we get our ribosome. And if we get our ribosome out in the cytoplasm, it's going to do the decoding for us. It's going to take the nucleic acid, the RNA, and make it into a protein. Well, if you're going to ship an RNA out of the nucleus, do you want to have to go searching all over the cell to find a ribosome? Probably not. You want it to be right there. So there usually are ribosomes very closely congregated next to the nucleus. So that leads us into our next organelle. Our next organelle is the rough endoplasmic reticulum, which sounds completely frightening, but it's really not so bad. Remember, in science, everything is very literal. The word tells you what that structure is. So this is endo. If you think of in, you think of inside. This is a structure that's inside of the cell. And it's plasmic. Well, we saw that with the plasma membrane. So obviously this is a bunch of plasma membrane located inside a cell. And it's reticulum. If anyone's kind of familiar with different kinds of animals and names, you hear reticulated.